So today we'll start with Christian Vestergaard from Pasteur Institute, and he's going to talk about mapping spatiotemporal dynamics of single biomolecules at the whole cell scale.
Okay, this seems to work. Thank you very much. Good morning, everybody. So I'm going to talk once again about single particle tracking, and so this is an approach that's very close to what David presented and what Pierre presented. And actually we also have a software package that's under development. So this is the competing software. You can find it at GitHub if you want to try it. It's still an alpha version, so much of what I'm presenting is not really implemented yet. So before I go into presenting the work, I'm just going to acknowledge people who did much of the work. So first of all, Jean-Baptiste, who's the PI of our lab, and who developed this mapping approach to start with that we're using. Alexander, who's a postdoc in the lab, who's a physicist. Francois, who's a computer scientist who's working all the implementation. And Mohammed, who did a lot of work that this builds on. So he's no longer working on single particle tracking. And I'm going to present some data from experimental collaborators at Antoine Trulle's lab at ENS, and from the labs of Delphine Muriot and Cyril Favard at Montpellier, and finally from the lab at Institut Curie of Mathieu Copé and Maxime Daron, who very sadly passed away this summer. So just give a quick outline. So I'm going to give a little bit of, I try to go over this quickly because this was already presented probably much better than me by David and by Pierre about the different questions in tracking single biomolecule dynamics. I'm going to present analysis pipelines. This is what we're working on. The idea is to have a fully probabilistic pipeline to make inferences robust and hopefully at some point automated. And I'm going to show some examples of the applications of this. And finally, I'm going to talk about a fun question that actually appears at the end of how to interpret the potential fields that we get out. And this is linked to the Itus-Tatunovic dilemma. For those of you who are statistical physicists, this may mean something. So first of all, a little bit, so very quick motivation. I mean, we've already seen a lot of applications of this. So a nice thing with these live cell tracking experiments is that we can link some molecular structure to the dynamics in cells. So we can look at biomolecules, we can change the structures we're using, for example, changing different base pairs, and we can then see what happens to the dynamics inside cells. Right? This is glycine receptor. It binds to a different scaffold in the postsynaptic membrane, and we can see that it forms so potential wells and gets trapped, gets stuck, the diffusivity drops as well. And if we change the beta loop here that interacts with gefferin, we can see that it no longer gets trapped. This is the kind of things we can see like in vivo, or in situ at least, dynamics of biomolecules and this to structure and also to function at the end. So this is for the motivation. So the kind of experiments that I'm looking at are going to be these kind of photo-activated tracking experiments. So basically, we have either SPT-PAM or UPAIN, which is just a different method, which gives more or less the same type of data. So we have molecules that stay visible for a very short time, so at high density, and the fact that we only have a certain fraction of these molecules visible at any time means we can get longitudinal measurements. So this is going to look, data looks something like this, right? So erratic motion, particles ping on and off, and from this we want to map so the physical properties of the membrane, so as experienced by the molecules that we track. So this I'm just going to list basically all the problems that I could think of. Try to go over this a bit quickly. So we have both intrinsic and extrinsic sources of complexities for analyzing these kind of data. So the intrinsic sources are basically what we can't do anything about. This is dictated by what goes on inside the cell, the physics of the cell. So the dynamics we're looking at is basically noise-driven. It's dominated by noise. It's random walks. The cells are both spatially and temporally heterogeneous. So they change over time, change with space. we might have multiple states of the molecules you're looking at, so they may change conformation, they may interact. We don't a priori know how many partners are interacting, what goes on really. There's no statistical physics that can tell us like equilibrium statistical physics here. And finally, we're actually looking at silicon cell experiments as well, because we're looking at measurements from single cells. So we have a lot of variability from cell to cell as well. This is a lot of heterogeneity that we need to cope with. Then there are those kinds of noise sources that are dictated by our experiment. We have localization errors. This was, I think, touched upon in quite some detail by Pierre. We also have motion blur. So the cells move during the acquisition time of the camera, so they get blurred. We can have mislocalizations. We can have mistracking. This is especially important when you have high density measurements. And I'm going to talk especially about mistracking. And finally, last, so as you say, step of complexity is in the inference problem itself. So first of all these errors accumulate through inference pipeline, so we need to take them into account to avoid having biased inferences. So from a statistical point of view, we have a large number of hidden variables. The inference is high dimensional, so we infer maps with, say, basically 2 or more parameters per cell, so we have a lot of parameters to infer, so this makes it complex from a computational point of view. And so this is maybe not really a problem, but we need to choose some trade-off between robustness of the inferences, so this is the precision, so by regularizing our inferences, and the accuracy of the inferences. If you choose less regularized, but then we get more noise. So that's, I think, all the problems. So now I'm gonna present the pipeline that we're working on to try to tackle at least some of these problems. So this is what I think almost every analysis pipeline of single particle data looks like. So this is a 3-step process. So we go from image stacks to localizations, using some sort of super resolution localization technique to get so sub-pixel, sub-wavelength localizations of the particles. We then need to link these localizations between the frames we have measured, between the frames we've recorded, in order to link this to the physical model for the motion that we have, that we want to fit, and then we can infer a model from this. So I'm gonna basically add probabilistic to these 2 steps. This idea is to be able to take into account all the uncertainties that arise here to make our inferences more robust. So I'm going to do this using a Bayesian framework. And the very nice thing, I think, about this is that this makes it straightforward to propagate the errors through the inference pipeline. And you can also do model selection in a very straightforward manner. So I won't actually talk much about model selection here, but this is in principle possible and should be in the future. So just to recap the basics, in the Bayesian framework, so as opposed to when we do frequency statistics, so we think of the data as fixed and our model and parameters as being random variables, and we can then build probability distributions of our models. And so a central formula, of course, is Bayes' formula that tells us how to go from a model of the data given probability of the model of the data given the model to a probability of the model given the data. So going back to the pipeline, I've now added probabilistic here. So at the moment it's all just formal. And if we just write this more formally, we have our data. This is the input data, it's an image stack. If you want to do localization in a probabilistic manner, this means getting a full posterior probability for the localization, so the super resolved localizations of the particles, giving the image data that we have recorded. Same thing for tracking. It means that we want to get a full posterior for the displacements, or the which is completely equivalent, the assignments of the particles, given these positions that we have super resolved positions. And finally, we want to get a posterior for a model and its parameters, given our localizations and our displacements. So if we write this all together, what we're really interested in optimizing is the marginal probability, so the marginal posterior, of our model given our data. So all of these goes in here, and then we need to marginalize over all possible values of the localizations of the particles and of their displacements that could have given rise to the data that we see. Right? So this is the proper way to do it from a statistical point of view. The problem is, of course, that we can't perform these integrals in practice because this is simply too hard. It's computationally too hard. So what we tend to do, and what is actually really done in practice, is that we condition on intermediary steps in the pipeline. So this is what we do when we do localization and tracking. So localization, instead of having the full posterior, we use the most probable localizations. Do the same thing with tracking. We use the most probable assignments or the most probable displacements, and we then infer our model based on this. And this works very well as long as these steps here give rise to unambiguous inferences. So, right, we need the probability distribution to be very peaked around the true values. So when this is not the case, this leads to biases in inference afterwards. So this is part of what I'm going to talk about. I'm going to focus actually on the tracking part. So for the main reason is that we don't have a working solution for this yet, for the localization part to give a full posterior for this. Second reason is also that tracking seems to break down first when we increase the density and when we increase fluorophore blinking. And finally, it's a more fun problem for a statistical physicist. But before going here, I'm just going to present the model. So this is exactly the same model as David presented yesterday. So the over-damped Langevin equation, also known as the Smolehovsky equation. And to infer this, we first need to make a spatial mesh. So this is the way we do it. This is a model. And inside this mesh, we then in each domain, we infer the forces or a potential field and the diffusivities here. So for meshing, some of the problems that we need to take into account are this spatial and temporal heterogeneity of the motion that we see. So basically, if you just plot all the localizations that we find here in a cell, we can see that there's like density variations, right? And the shape is, of course, not regular. So we need to take this into account. And we have implemented, like, for the moment, 4 different methods. So the first I'm not showing, this is just a regular grid. This doesn't work very well. And the problems with the KD tree here, so this allows us to adapt the size of the domains to take into account a differing number of, different density. K-means is also a very standard method, which allows us to adapt the shapes here, but it doesn't allow us to adapt the sizes. And so François, our computer scientist, he devised a new method based on a growing neural gas that actually gives us the best of the both worlds, in the sense that we can get both adaptive sizes and also adaptive shapes. Right? And we need to also test latency in time. And for the moment, we just use sliding windows. This is easiest. And as I think pointed out by Daniel yesterday, there is a fundamental trade-off here again between the resolution that we take and the statistical resolution that we get out. Right? The larger the bins, the higher precision we get in the bins, but the more bias we get due to the fact that we may not get all the variations that are actually there. In any case, what we're going to do after the tessellation when we infer is that we're going to regularize between bins to get smooth inferences. So the physical model we're using, so this is the Langevin equation, or the Smolehovsky equation. This gives us directly the probability for displacements. So this is written as function of diffusivity and a potential field. And we use a regularizing prior here. This is, again, a nice thing about the Bayesian framework is that we can put regularizers directly as priors. We simply just impose that we want to have some smoothness between cells of of the solution.
Can I just check? Is this
Yeah.
the model theta that you want to fit it?
Yeah, this is theta. So actually we're only... Oh, sorry, yeah.
Yeah.
Okay, yeah, so we're only considering one model here. There's not going to be any competing models for the moment. So the model is this, yeah. This is theta. And so theta is the model and its parameters. Yeah. So our model is Langevin equation with v of r and d of r. Right, there's going to be 2 times the number of domains is the number of parameters.
So just to clarify, is this the Langevin equation? It looks like the Stratanovich form, which usually people use when they go backward in time. For example, this has been used very much in the Zakai equation for signal processing, where usually it's not physical. You have to have the information about the future, and from the future you go back in time. Is it the Stratanovich form or is it the Ito form?
It's actually the general form. So if you have the Ito form, you don't have this lambda delta d.
Okay.
So this. So the reason I put this here is what I'm actually going to talk about in the very end of the talk. And the idea is depending on how you choose your integral convention. So if you use Ito, I should say Stratanovich, or even isothermal, sorry, convention, you're going to get different. Actually, you're going to get different terms appearing here, like in the Fokker-Planck equation and the solution to the Fokker-Planck equation.
Okay.
And the idea is if you put a lambda, this just gives you all types of conventions.
Okay,
principle.
but you know that they are unphysical. There is only the Ito. The Stratanovich is not physical. I mean, this has been debated 20 years ago already in the field that you should not start with the overdamped. You usually start. I mean, you have to start with the Langevin equation, and from this you go to some limits. And it turned out that Stratanovich isn't physical, and so that's why people stayed to that day to Ito, unless you do a signal processing, but it has nothing to do with the physics. And then they used it in signal processing, and you can read, there's the entire second book of Schuss dedicated to this question.
So I don't completely agree. I must say, so the thing is here that, so if you take this as a limit, yes, you say as a limit of going into the overdamped regime. So depending on what your temporal correlations look like, you can get different solutions, right? The Stratanovich will actually come if you have a symmetrical, so a time symmetrical correlation function. And the Ito, as you say, this is the one that doesn't look into the future. And it's actually also the one that's going to be most, by far most practical when we want to integrate this equation, because we don't need to look into the future. But if you want to, actually, if you want your solution to respect Boltzmann equilibrium, you need to use the isothermal formulation where lambda is equal to 1 here. So Stratanovich would be 0. So 1/2, Ito is 0, and isothermal is 1.
You don't have Boltzmann distribution if D depends on R. It doesn't work. If D is X dependent, you don't have exponential. If you cannot solve the equation. If D is constant, that's true, you get the Boltzmann. In general, this is discussed in our biophysical review in 2014 or 15. You cannot have it.
I think there's been a lot of discussion of this. I don't think there's any...
and again, this has been discussed over the 20 years, not in the physics literature. This has been discussed mostly in signal processing, Wong-Zakai equation from the Technion, who developed all of this. And you should look, there is a book, 2012 in Springer, discussing all of these issues.
Okay, I'll look at this. I don't know about this. I know about a lot of discussion going in the physics literature, and also from Kampen, for example. He actually says that you shouldn't use Langevin equation at all because you don't know what it corresponds to. Our point is here that since we're in biology, as you say, we don't know whether equilibrium should be satisfied or not, really anything. So lambda could be anything here, from what?
Yeah, if lambda is 0, that's
Okay, I think maybe we'll discuss more later because I don't think we're gonna reach an agreement right now. So that's the physical model, and this is already not clear, right? What to put into this, and a second thing that comes in. So these are now the extrinsic noise sources. We have localization error, so it's pointed out by Pierre, and also motion blur. So it's the second part. So basically, we leave the shutter open for some time when we track the particles to get enough photons to be able to localize. And so since the particle moves during this time, we get some blurred position. And both of these not only impact localization precision that we can get, they also impact the statistics of the time series that we see afterwards. So this is the variance of the displacement. So this is the typical diffusion. So 2 times the dimension times the diffusion coefficient, and you get you get a term here that depends on localization error and motion blur, and you also get an off-diagonal correlation term, right? And this complicates matters a bit, the off-diagonal term. So what we're going to do is we're going to ignore it. This is what is usually done. So this gives us the likelihood that factorizes. So it's going to make it much easier for us to analyze things. So the good thing is that when we ignore this, it actually corresponds to using a moment matching estimator that was described in my paper here, and this actually works very well in practice. So as long as we have a signal to noise ratio that's larger than 1, we get practically optimal results just ignoring this.
If you ignore the covariance, you're only doing single particle tracking at that point, right? Because once the PSFs start overlapping, you can't deal with that issue anymore.
Well, this is actually after localization. Right, this is what you get out after. So this is like the statistics of my time series once I have localized.
I
so
see.
for doing localization,
but I guess this would, I don't know. I mean, if you already pre-localize, I'm not sure how this really helps you. I mean, the difficulty is, I guess, is localizing once the PSFs overlap, right?
No, they don't overlap that much actually.
Okay, so you're doing basically primarily single particle tracking.
Yeah. Okay, no, that's not. I mean, the idea is we want to have single particle tracking. So maybe this was not clear. Yeah.
No, you can't have many particles overlap. That's my point.
No, then it gets complicated. Yeah, exactly. So the density of some of the densest images you look at is around 1 particle per micron. Right, so there's not going to be a lot of overlap, but it's still enough to create some problems in localization, especially tracking. But it's not like you don't have to deconvolute more than 2 particles overlapping at one time. So I don't know how much. Okay, half an hour. Okay. So this is just to show. So this is like pure numerical, right? So we can also recover temporal evolution of these maps. So if we have a diffusivity that changes shape over time, we can recover this from the dynamics already.
Can you explain a little more what's being simulated?
Yeah, actually, I mean, we simulate. So I didn't show the simulated maps, but basically they look like these. Right? So we have a diffusivity well that moves over time, like this. Right? You say a throw, right? Diffusivity decreases in the center. And you can recover this just from the dynamics here. So now I get to the tracking problem. So when we get to high densities, or not as high a density as what Steve was asking about, but high enough density that path of the molecules would tend to cross, we get a problem with linking the particles positions. And this is what I'm gonna focus on now. So here I'm just showing a simple example. So what happens if you just have 2 frames? So on average, 33 particles per frame, and we do tracking, and then we infer the diffusivity. So this is a function of the density, and what happens is, so we compare this to if we know the true trajectories. So this is the best we can hope to do. But if we don't know the true trajectories, we'll tend to get biases whenever the particles cross. And this happens more and more often as the density goes up. Right? So the most probable trajectories are actually not the true ones, because the most probable is going to be the shortest ones. Now, when we also add photo switching to the equation, the things become more complicated. So we have just put everything on the same scale. And especially when we get a lot of blinking, so we'll tend to get a lot of mislinking now. And this actually causes a really big increase in variability, and also can cause biases like in both directions. And so this is more or less where we are with like some of the high density experiments that we got from collaborators. So actually, we do need to take this into account to get more reliable results. So to do this, I'm going to, instead of using basing or inferences, as I said, on, yeah, sure, yeah.
Can you go back to the slide before?
Sure.
So you're mostly showing overestimate of the diffusivity for smaller time delays. What's that?
The colors maybe not very good. So this is the median, see and this is the confidence interval.
Oh, I see. So the darker band.
The darker band is what you get if you know the true trajectories. So this is like the best you could hope to do.
Okay.
Right? This is just statistical error then from the fact that you only have 33
No,
particles.
but the darker purple is the
yeah, so
median?
the purple band is going to be the 95% confidence interval.
Got it. Okay.
And yeah,
But
the line in the middle is the median.
Got it. But over a big range, you're more likely to overestimate than to underestimate, right? Is that just because when they blink, then you link to a particle that didn't blink farther away, and so you think you're making bigger steps than you really are?
Exactly, yeah.
Yeah, okay.
Yeah, this is what happens, right? If this one goes off, blinks off, say, okay, this is not what I've shown, so actually, but this is what happens if something appears out of nowhere and something else disappears. So you tend to link then these particles that have appeared or disappeared, and since they are further away on average than the actual jump length, you will tend to overestimate. And this again is of course depends on the density. So as we get higher density, you actually go back to underestimating.
Okay.
Right. And but this is much higher density than what we see in what people can actually give us in experiments. Yeah.
Sorry for all these questions. We're really slowing you
No,
down,
no.
but the two things. So how are you doing your tracking? Are you using IC or something like that?
Oh yeah, I didn't say that. So what I'm doing is what I call this the most probable assignment. So this is really, since I know exactly what generated this is MC simulation, so I know what generated this, I use my probabilistic model. So I take just the most probable assignment. So this is supposed to be the best.
Yeah,
right?
okay.
If I use another tracking, it's going to be something. I didn't want to compare to, because there are 1,000 tracking methods, and I didn't want to say that one is bad and one is good, or yeah. But actually, I mean, of course, this is what you get is going to depend on what you track. The real main point is that you don't know what you're going to get if you track. So this is, I think, that's a real problem.
And just a second question. So when you're doing your Berglund correction for the errors, are you throwing away only the off-diagonal terms and keeping the correction to the diagonal, to the variance?
Yeah.
That's what.
So otherwise, you get a bias. Yeah.
Yeah. Okay. Yeah.
Yeah. Okay. So this is like the technical part. So what we want to do is we want to calculate the marginal likelihood, the full marginal likelihood, instead of just relying on tracking. So S here is a given assignment. This is the tracking. So just showing everything here. So we need to sum over all possible numbers of particles that can have appeared and appeared, and we need to sum over all possible assignments. And this assignment sum is the problem because this is so actually not NP hard, it's sharp P hard, so even worse. And the way that I'm then going to do this is to map the problem to a graph matching problem. So each of the terms in this sum is actually completely equivalent to a perfect matching problem on a bipartite graph. And calculating this inner sum is then equivalent to calculating the partition function of the graph. So this is a link to statistical physics, and we can then use the belief propagation algorithm to solve this approximately in polynomial time instead. So, okay, I'm going to skip a little bit quickly, but basically we write the likelihood of the matching as a factor graph. This is the factor graph. So belief propagation basically works by iteratively sending messages between nodes here about the belief of each node, about the state of the other node. And so the nodes, they update their own belief about their state from this, and this will give us the marginal probabilities for the state of each node at the end. Right, so the messages look like this. And then we get the minus log likelihood is equal to the Beth approximation to the free energy here. So it is the free energy, and we use the Beth approximation. So this is not completely rigorous, it's an approximation, but it works very well in practice. And the nice thing is here that we can implement this in a parallel way, because this is basically just matrix multiplication. So the final thing that we need to put in is to estimate the number, so distribution of particles blinking on and off, number of particles blinking. And so we're going to use assumption of stationarity, spatial homogeneity, and this gives us directly the shape of the distribution that we can fit to real data. So this gives us the hyperparameters that we need, the parameters that are that gives us the distribution for how many particles are going to appear and disappear. So we can put this directly into our non-tracking algorithm. And when we do this, so this is non-tracking, we now recover results that are almost as good as if we actually knew the true trajectories, even at high density and high photoblinking. Yeah.
So when you say you're assuming things are spatially homogeneous, are you assuming that also about the diffusivity?
No, not at all.
No, so yes, because it seemed like spatial variation diffusivity is what you want to study. So what's spatially homogeneous?
The photo.
Just the photophysics.
Photophysics. Thanks, that's the word I'm looking for. So the photophysics are, I assume they have to be more or less spatially homogeneous, because otherwise it's going to be difficult for me to treat. I don't need actually stationarity, because we can also fit it if it changes over time. But constant rates and homogeneity, I assume this. But this seems to be pretty, a good assumption. And it's not overly, sensible to like too small changes in this. So I'm actually quite soon running out of time, I see. So with this, I'm just going to show quickly some example applications. So first here is some non-time varying maps. Actually, where I don't use a non-tracking, I use normal tracking because the density is not that high, and also GAC protein dynamics in the formation of varied capsids, where we have temporal evolution of the maps, and where we also use actually a simplified form of the non-tracking algorithm. So first, the glycine receptors. Just the motivation, right? This is linked to synaptic plasticity, synaptic strength, how information is processed in the brain. So we want to know how, say, receptors interact and move in the membrane. So these particles, tracking experiments, so gives us data something like this. So the white dots are the glycine molecules. And we get maps like this. So potential map, diffusivity map, and we can see these potential wells. And we can also see that we can have different diffusivity wells as well here. So this depends on actually the specifics of the molecules going on. And so a nice thing what we can do is now we can link, so again, structure, dynamic, and function. And if we look at here the glycine molecule, we can go ahead and make different point mutations. So for example, in activating the last part, you have the beta loop that interacts with Jefferin. So this is the red points. And we see that this basically kills all the potential wells, so there's not going to be much interaction anymore. We can modify it a little bit, so this makes it less active. And this actually doesn't change the potential well that we see, but it changes the diffusivity inside, so it makes it less stable, right? It gets less immobilized inside these adhesion points. We can also go in and make functional studies. If we treat the cells with a phosphorylizing agent, and we also look at different here other point mutations, we can see how this changes the interaction potentials, the diffusivities inside inside the synapses. Okay, very quickly. So these GAG proteins are linked to the formation of capsids, so especially in the HIV virus. So these form like and bud on the membrane. And here we have time model dynamics. We're going to look at 20-minute recordings because this happens over, say, 15 minutes. And again, we can do different kinds of functional studies here. So change the role of the catalysis or change the interactions between the different molecules. And we can then go in and make these time evolving maps. So here we see, this is the density. We see that something's happening here, right? We get interaction. And then we see how does the diffusivity and the potential form and change over time. Right? And we can plot this on a time axis. We see density going up, diffusivity going down, energy going up, going down actually, right? This is the depth of the potential well. And we can then go in and study actually, so with different modifications, we can see how, so changing the interactions between the molecules will change the depth of the potential well. It also changes actually the synchronization of these different events. So it's actually quite orchestrated when each thing happens. And we can see that we can actually make it both, this is the wild type, more synchronized or less synchronized by changing these different things. So this is a delicate balance going on of stuff here. So this is the final point, which David also referred to. So the question of how can we interpret these maps when we have a Langevin equation. So I don't think we're going to reach any agreement on this. But the basic idea is, so now we write this here in Ito form, but with the integral convention not specified. So what the idea is, to what extent are these drifts that we measure, can they be explained just by varying diffusivity? So if you use Fick's law, this is equilibrium physics, we know that we expect some drift just due to varying diffusivity. But if you use the Ito convention, there's not going to be any drift. And the idea is that since we're in biology, we don't actually know anything about what are the statistical physics of the system. It's complicated. So if we don't know what, this lambda is, I didn't put it here. So if we just use different conventions to do our inferences, we actually will get different results for these force fields. So we can get different results. So what we can do instead is take the same approach as for the non-tracking. So we just marginalize all possible conventions, and we get sort of a more conservative estimate of our forces. And we can actually use this to say something about when we measure these force fields, are they solely there due to diffusivity, or is there actually something else going on, some active forces? This is for, again, for the GAG proteins. So we measure a drift field. Okay, this is quite noisy, but we can see drift fields. This is a spurious force generated by the varying diffusivity. And here we can so basically calculate a base factor for the evidence for an active force over the null hypothesis that there's no active force. We can see that at these interaction sites there are active forces. So that's it. Oh. So this is the probabilistic pipeline, so it is not completely finished yet. So the idea is that there's a probe these complex interactions between biomolecules in situ, and we're also working on some. So this is work in progress. So trying to have a generative model for localization to make this also probabilistic. So we're using variational autoencoders. And another fun problem is actually trying to use different models. So not only the Wilhelm-Langevin equation, but using actually going beyond what you can say the canonical models of motion, which are fractional baryon motion, Langevin equation, continuous cerebral walks, etc., and try to go away from trying to distinguish between all of these and actually see if we can use a mixture of these models, because in biology probably our simplified physical models are not going to describe what goes on. It can work on technical of equilibrium dynamics in these noisy trajectories, and so the goal is to get like an automatic analyzer at the end, a biomolecular analyzer. Okay, that's it. Thank you.
We have time for one question.
So it remains very unclear for me. What is this, your map or your energy map? In physics, what is important is the difference of energy between 2 points. That's how you calculate, you know, from 2 points you calculate different energy, and this give you forces. Can you come back to this map that you have? And it seems that you can go in your map with like, yeah, for example, with high energy like 8 kT in some very large region, and then you go to changes. I mean,
this is just zero. Is just. I mean, this is just the scale as not the scale. Sorry, the where you put the zero, right?
So you assume, so you fix, because the energy is defined up to a constant, right?
Yes, you're right.
Okay.
Exactly.
So I assume that zero is at the end of your potential.
Yeah, exactly.
Like okay, but
this
still,
is just a representation. I mean, it doesn't.
Yeah, no, but still, a representation is important. This is how we visualize things, and there are regions where you have quite significant changes in the energy landscape. And do you have any idea what that means? Because you see, in the potential well, I understand. You have a local structure, you can have a difference of potential. I mean, we thought, you know, it may be a mixture between electrostatic interaction and something more occurring at the membrane that it seems to be important in many cases. But in other places, why you would have this difference in energy that would be associated to the first moment, which means like a force, like something. What is your interpretation of this?
So of course, the true answer is that I don't know what's going on. And the slightly longer answer is that since this is biology, I mean, things do really tend to interact over long distances and change things. Actually, something that we do know, here in the VLP, what happens is that as these gag proteins, so these are the sticks inside, they bind, they will deform the membrane. So this will actually change really the potential energy, right?
No, this I understand at this scale, but you are showing other scales which are longer. You know, it's in your entire like few micrometer, no?
So, well, the question is whether
it's. I understand what you're saying. I perfectly agree with you at the level of. We had also this data last year where we looked at this variant.
Synapses or the VLPs?
We both at this gag and we also see indeed locally. I agree with this, but further than that, you know, the interactions actually for a cell. I'm not sure you would like to have long-range interaction. You would like to have fast trafficking at least to reach the spot, for example, at the synapse where you need to have this specific location. This makes sense, but away from that, it's not clear to me what is the underlying. Our job is not to say that, okay, I understand it might be some biological structure, but our job is possibly to make sense through the physical, you know, understanding.
I agree. So I know I can say that I don't know what's going on. So part of this can also be simply artifacts, right? Statistical, because I mean, since we impose regularization here, right, to get smooth structures, of course, there's going to be some remnants in between. Yeah.
Okay, let's thank Christian again. Thanks.
Perfect. Now do you hear me? All right. So thanks for inviting me to this great opportunity. My talk is going to be a little off from the other talks that I heard. It's confocal and not imaging. It's mostly in vitro, but I hope I would convince you that still it's kind of interesting. And 2 talks ago, we heard about confocal and time resolution, and I touched this a little further. with my technique. So basically, everything in biology, all the processes rely on macromolecules, protein, DNA, RNA, macromolecules, but specifically on their structure and even more specifically on their temporal structure. Different macromolecules attain multiple different structures and more specifically, multiple different conformational states, where each conformational state carries on a specific function that is decoded in the structure. Binding of a specific molecule, enzymatically changing that molecule, transport of an allosteric signal, you name it. And so therefore it's important to really define those structures. And indeed, you have the whole field of structural biology. Many decades of crystallography have allowed us to gain access to atomic resolution, three-dimensional structures of many proteins in many states. But still, I think the PDB, largely saying, is sparse on another dimension, on the conformational dimension. Since in crystallography, you usually use, you create a crystal with organized molecular species in it, with certain biochemical methodologies. Usually you capture the most stable conformation in your solution. Sometimes even you induce a stability that wasn't there from the beginning in the solution. So, and there are many other conformations that might have been ignored in this process. And here's an example of adenylate kinase, where this is just, a fake movie because it morphs a lot of different crystal structures of adenylate kinase. But there are many others that weren't defined, but still in single molecule experiments were kind of identified. And here is an example of an experiment from 2007 that really have shown that even without a substrate that induces these movements, the domain move. They move in a large scale movement, and they are also coordinated in time. So this is pretty interesting. But this is also important because, well, you may say this is a huge macromolecule with a lot of atoms, a lot of modes of motion it includes, from very small scale bond rotations to very large scale domain movements. But still, you can kind of reason on energy landscape concepts, where you have an ensemble of a lot of different structures that are closely related in a potential well, where you can move between those closely related structures in a diffusion-like process, so it's less than KT transitions, or move between different conformational states, different potential wells in an activated process. So largely speaking, we can try to identify these conformational states. You can see that they might have different activation barriers. So the idea is of integrative structural biology is to try to use our other techniques, other biophysical techniques, to identify signatures of those different structures and to classify them. And indeed, nowadays you have a lot of different techniques that are being used for that. The most famous one is structural determination from cryo-EM experiments. Eva Nogales a few years ago have presented classification algorithms that allow you to differentiate atomic resolution structures of different conformations. She has actually reached, I think, a 20% abundance of a conformational state in the whole frozen ensemble of different structures. So nowadays, with good computing compared with cryo-EM imaging, you can really reconstruct conformational states that have overall different structures. And but you also have other techniques like NMR and EPR, which are really ensemble methods. And MASPEC with cross-linking, that with good computing you can reach the idea of finding the heterogeneity of the structures. But what I'm going to talk about today is how you can use single molecule FRET and single molecule fluorescence-based techniques in order to reconstruct conformational states. So just a brief remind for what is first resonance energy transfer. You have two dyes with a spectral overlap between them. One is behaving like a transmitter, a donor. The other is acting like a receiver, an acceptor, both of Coulombic interaction. It's a weak coupling between those two dyes, basically between those the dipoles, the excited state dipole of the donor and the ground state dipole of the acceptor. And after you do the calculations, you basically end up with the Förster mechanism that states that at least instantaneously on a frozen molecule with frozen dyes in space, the distance relates like the sixth power dependence. The distance dependence on the efficiency of the transfer, meaning the amount of photons you get in the acceptor divided by the amount of photons you get both in the donor and the acceptor. So, and that's basically the contribution of Theodor Förster, but also before that, other great physicists, for instance, Perrin, found it with different physical theories. And later on in the 60s, these two guys really made it an experimental molecular ruler. And since then, the field is thriving. But another addition to the field was that until the late 90s, people were using this technique in order to measure an ensemble of molecules, all unsynchronized, to getting an ensemble average of the distance. Thinking that, assuming indeed, like crystallography guys think, that you have a general conformational state with one mean distance. So you will have a one-to-one mapping between the ensemble average to the ensemble distance, but we all know that's not true anymore. In single molecule measurements, what you have, at least with a confocal spot, is a molecule that is doubly labeled that crosses by diffusion the beam. It's a confocal beam. It's tightly focused. So the overall intensity of the beam is very low, which makes the effective excitation and detection only at a very small 1 femtoliter detection volume. So the molecule crosses by diffusion. Of course, this is not a random walk type of diffusion, but just for illustration, crosses the excitation volume, and then it creates photons that are detected, and they are highly bunched in time in comparison to the process of the background of the detector. So you have spikes. And the lower your concentration is, the better the different single molecule that you measure are resolved. And the better you resolve also the signal to background ratio, which is important to the detection. So you can take ratios of photon counts in the two channels, red versus green and red, and you can get for each burst the FRET efficiency, and you can quantify them in the histogram. So for instance, here in 1999, it was shown that you can basically measure a DNA, a mixture of them that have different pairs of labels at different positions, where you have high FRET, which is bunched here, low FRET, and a donor-only one that can either be synthetically like that or produced by bleaching. And that's basically my postdoc PI. I've been working with him for the last 5 years. Just lately I relocated back to Israel. So in 2005, Shimon introduced a new technique that instead of shining just one laser to excite just the donor and observe donor and acceptor photons, you, in the microseconds time scale, alternate between either donor excitation or acceptor excitation. And that gives you another dimension, because you can measure not only the FRET efficiency, that is all dependent on the donor excitation period, but also something called stoichiometry, which is the overall photons introduced in the donor excitation period to the overall photons introduced both in the donor excitation period and in the acceptor excitation period. So then you might have molecules that didn't have an acceptor at all. So they will have a stoichiometry of 1. Molecules that didn't have a donor at all, they will have a stoichiometry of 0. And molecules that had stoichiometry somewhere between 0 and 1, which means that the fluorescence of the acceptor is active, you can better resolve stuff. So you can do things either with confocal-based microscopy, but then what you really get is information on single molecules just for snippets of time. Okay, because the molecule diffuses through the beam in 3 dimensions and then is lost. And it diffuses roughly at the 1 millisecond time scale. Some do more, but most of them diffuse in 1 milliseconds. But on the other hand, you can also image them with TURF, for instance, or lately you can use avalanche photodiode arrays, like you mentioned. Xavier Michelet should have produced a talk, and he wasn't able to arrive, so I just mentioned that. But on the other, so you can really focus on single molecules for a very long period of time until they photobleach. But on the other hand, the usual cameras that are used rely on binning, and the latest scientific CMOS really allow you to see enough photons in a bin that you have a time resolution of around a few milliseconds. But avalanche photodiodes used in confocal optics allow you to see, they have a dead time of detection of around 100 nanoseconds, and it's getting even better. Which means that, in principle, if you have bunching of photons, you can see dynamics as fast as a few hundred nanoseconds. So, and there are other tools that are also molecular rulers, some of which I was involved in developing, some of which are not. For shorter distances, lately for imaging longer distances, for instance, the distance of a surface of a cell from the cover slip. So I will briefly talk about PIFE, where basically instead of just having 2 dyes, 1 dye is sensitive to the environment. So you sense not only the changes of the conformation of the DNA with these dyes, but also whether it's because of protein binding or just because it's intrinsically moving from low to high fret. You have another thing called quenchable fret with 2 specific dyes that when they are too close to each other in contact, one quenches the other, so it introduces a donor only temporally. So here's an example from the literature, and it's ever growing now, of what people have done with this technique. So fret is a 1-dimensional reporter because it reports just on a distance in space. In order to get the 3-dimensional information, you have to do it for many times, for many reaction coordinates at many angles. But look, this protein already from the PDB is known to exist in at least 3 different conformations. And indeed, doing this mapping, actually a lot of experiments have been put here, basically maps back not only the structures, but also their abundance and rate constants. Yeah.
What's atlastin?
So atlastin, and I think I heard a lot about it in the last few days, is one of the proteins involved in the lumen of the ER, right? I saw it popping there. So this is not my focus, but I just wanted to show you a very nice example of the 3-dimensional reconstruction that you can get out of multiple 1-dimensional information. But still you have a time resolution problem, and here is the reason why. So I mentioned that the diffusion of a molecule through a confocal spot is usually at the time scale of a 1 millisecond. However, the transitions between the different conformational states associated with different fret and different distances between dyes might be faster than that and still be considered as activation barriers that are larger than KT, at least for proteins intramolecular changes. So in principle, you might have those 2 states bunched into very fast interconverting states that look as if there are just one subpopulation, right? If you rely not too much on photon statistics, but just on building these Fret histograms. So that's not too good. We want to do better. And there is a plethora of techniques that basically allow you to do some calculations on photon statistics to get better resolution, the time resolution. One of them is FCS that was mentioned here, where in FCS you really rely on time separation between different photophysical processes. Sometimes you don't have that. Here it's depicted as hundreds of nanoseconds for Fret, but it might be slower than that for Fret changes, and then it will be mixed with triplet processes. So I agree with you, FCS is not the best approach. One approach I like, still qualitative, but it's a single molecule and direct approach, is a burst variance analysis. So burst variance analysis is the following. This is just bin data. What you really get is is a sparse time series of green and red photons that you can basically, let's say that you have 30 photons here. Okay, and you can think about it, if it's one conformational state, then choosing whether you are going to get an acceptor photon is a Bernoulli trial. Okay, it's basically a binomial process for each photon, and for all of the photons it's a Bernoulli trial. So you would expect the standard deviation of that, or the variance of that, to behave like the value that you have inside the root here. So you can take each burst and calculate not only the instantaneous Fret, but also the standard deviation of it by bunching the photons into consecutive constant number of photons, calculating the instantaneous ratio of red to green and red here and here and here and here, and then taking the standard deviation of that. And what you will get is usually bursts that circle around the point which lies on this semicircle, which is this. But if you have molecules that, while crossing the beam, moving from a state with more red photons to a state with more green photons, so you have moving between bunching of red photons and bunching of green photons, what you will get is an overall larger standard deviation for those bursts, which is directly a signature to show that if you have a single population here, it's not from a single species, from a single conformation, it's from more than that. So, and here is a demonstration. This is just a simulation to show you. Here you have a single state that was simulated, another single state, a mixture of two static states, and states that interconvert with rates that increase until you see just one population. So it looks, for the experimentalist, or at least the one that just uses Fret histograms, as if there is one species here. So indeed, for a true ground state, one state, you have all of the bursts really aligned on the semicircle. Same goes for the two states. But as you reach this situation, here you have this bridge, bursts that are in the bridge between the populations that just interconvert while crossing the beam. You see that as data that goes above the semicircle. But even in the fast case, you still see remnant signature for dynamics for something that obviously looks like as if it's just one state. So I was using this technique to start my classification. My scheme of working is as following. You have a lot of cards here. Each card is just one experiment probing one pair of dyes at a given set of positions. So it's a reaction coordinate if you wish. I get a Fret histogram, and let's say that Fret histogram includes one centrally distributed population. I wouldn't say a Gaussian because it's not exactly a Gaussian. Then I ask, is this one state? To answer this question, I go and perform BVA. If it's just static, static heterogeneity, which means that the data points for that population lie on the semicircle, I move on and extract the distance information from it. If not, then I have to quantify what I saw qualitatively as heterogeneity of states in something called photon by photon hidden Markov modeling. So after I reach the knowledge of all the distances of all the conformations, again I ask, was this one state? Then I don't need to classify it because it was one state. But if there are 2 or 3 conformational states dynamically interconverting, different measurements of the distance might mean relate to the different conformations, and you need to classify them back. So we will reach that point later on. Very fast forward, photon by photon hidden Markov modeling is hidden Markov modeling on the bunching of photons, basically on the probability of, if you have a red photon at a given region in time, what will be the probability of the next photon to be red or green, depending on the density that of green and red photons that you should have per a given fret efficiency. And this is just an illustration to show you that indeed you have here a region, at least in this illustration, of a lot of red photons, and then abruptly it changes to a lot of green photons. It's not that you don't have any more green or red photons, but the density is really reduced. And this relates back to the idea of a Bernoulli trial for the bunch of photons that you have per state while the molecule was at that state. So, and you know, the technique has been proven by these guys in the Weizmann Institute, Menachem Pirchi and Gilad Haran, very well for a molecule, a holiday junction that is well known. It has a prior. It's well known to be interconverting between 2 states. And I thank you for introducing the idea of model selection, because really the problem is, if you're more exploratory on your molecules, the question is, how many states does your HMM model describe? So there are many estimates of that relying on the minus log likelihood, which is here. One of them is the Bayes information criterion that tries to weight the log likelihood with the number of parameters and the number of data points you have, where Q here is the number of states and the number of free parameters you have for a given state model is the number of states plus the number of overall rate constants that you have. So, but basically the problem is that when you compare between different measurements, you have different number of photons. And if you want to compare between different measurements, that's a bit problem, because then you will have a different minus log likelihood, different Bayes information criterion, different everything. So what I just introduced, I read a bit of literature on the statistics theory of Bayes information criterion, and basically the idea is that you need to reach a model with a minimal number of states from which on the Bayes information criterion is redundant, so it produces basically the same estimate or even worse. At my case, it was at around 4 states. And the number of photons is huge. You basically rely on all the photons of all the bursts you accumulated. These are tens of thousands of bursts. Each of them contains somewhere between 30 and 100 photons. And what I did is basically I took this delta BIC and normalized it to the number of free parameters of degrees of freedom. And what I saw is that while different measurements, or here, different simulations produced different values, where I really want to have a standardized cap on which is conceivably statistically meaningful and which is not, for this tag BIC, what I get is I can introduce, let's say, a 0.005 threshold, and it will work for all of my measurements independent of how many photons I produced, independent of everything. So basically what I say here, let's say for the simulation of a two-state, is that one state doesn't work, two-state works, and 3 and 4 of course works, so you stop with the...
How did you, I guess, how did you derive the BIC prime?
Oh, so you have the series of photons?
Yeah, but the formula, the formula itself divided by n minus k, how did you derive that?
I have the BIC.
Yeah.
Okay.
Yeah.
And I have the BIC per a given model.
Yeah.
So, and I also have the minimal BIC out of all the different model comparisons I did.
Okay.
All right. And I called it delta BIC. And in the literature, actually, it's also like that. Then I just take this value and divide it by n minus k to account for the fact that when I'm comparing different experiments, the estimation of a given state model for different measurements, I will have different number of photons that clearly show up in the BIC here as different values, even on the simulation level.
But I guess, fundamentally, there is that log n correction in the BIC, right? So the BIC does tell you that the number of parameters
L
grows.
is minus log likelihood here.
No, but on the right-hand side, you have k log n. Yeah.
Oh, yeah.
Yeah, so it does say that the complexity grows logarithmically with the number of data points. So, of course, the next 1,000 data points compared to the first 100 will only produce as many parameters as the first 100. So there is that factor already built into the BIC. It's a rigorous derivation.
Right.
But I feel like now you're...
This is good if you're comparing different models on top of the same data set. But if you're going to compare...
this is different size data sets. The log n is specifically to account for different... It's an asymptotic result that accounts for different size data sets specifically. That's where you have that log n correction.
So
I think you might be...
I read the same literature as you did, and I expected that, but when I went on and compared BIC, even on the same data set for different models, I got different values.
I think it's because the k might not be... There's an interpretation of k. You said k was the number of states and the number of kinetic parameters, right?
Yeah. Basically, the number of parameters that you have is k. It's the number of states plus all of the rate constant back and forward.
I think it's only the rate plus the emission parameters. I don't think it includes the states. And I think it's the number of
emission parameters are the states.
State. Well,
but the emission parameters are the states, because when you have an emission rate of green and red, it defines you the state in means of the fret efficiency.
It's going to be multiplicative with the number of states. Yeah. But it might still be different from, so therefore your k might be a k tilde, and once you incorporate that k tilde in there, you might not need that additional divided by n minus k recorrection, I would think, maybe.
Maybe. We can talk about it later. I'm really interested in improving it. But what I did is basically used simulations in order to at least convince myself that the ground truth is recovered by this. At least when I looked just at base information criterion, it didn't. And it's clear from the fact that it fluctuates around different values, and I can't, know apples from oranges.
Well, there's an asymptotic correction, I guess. Well, sorry, I don't want to interrupt too much, but there's an asymptotic correction to the BIC. BIC is only leading order, so you do the first order in Laplace's method, but then there's asymptotic corrections that scale as 1
does
over
it work
n or
also
1 over n
on?
squared.
Does it work also on hidden Markov modeling inference of rates?
I mean, I don't know if it has been applied, but it should apply. So I think if maybe adding those additional correction terms might also give some kind of improvement as well.
I would love to hear about it later. All of that was just methodology. What I'm focusing on is the transcription initiation in bacteria. I'm doing it in vitro for various reasons. You can highly control your system. You produce a lot of data, which for me is like a dictionary that later on you can go to the cell and try to find signatures that you already found in vitro. This is like an inverse approach of most of the talks I heard, and it's okay. So what you have in the cycle is the following. You have what's called the core RNA polymerase, and the sigma, which really is the specificity unit that identifies the promoter of a gene. it can't identify the promoter of a gene alone. Only upon binding and creating the holoenzyme, it can start functioning in searching the promoter. Upon binding to the DNA at the promoter region, the DNA is bent, and throughout a few stages, there is melting of 10 to 12 bases from -12 to +1 relative to the transcription start site. Okay? And I said 10 to 12 because there is also transcription start site heterogeneity that I will talk about later. So after the bubble is open, and active site here, it's an illustration, but basically the crystallography shows the same thing, just more complex. The active site here is constructed out of a few amino acids that coordinate a magnesium and the incoming nucleotide in order to polymerize it to the previous nucleotide. So only when you have this space it can occur. But you have also a constriction coming from the sigma factor that blocks the channel from which RNA is exiting. And basically this block has to be removed in order to move into elongation, and this is basically why initiation is so slow and so rate determining. Most of the regulation is occurring either in initiation or in termination. So what happens while the constriction is not removed? You have a lot of abortive short transcripts that are produced. These are simply failed experiments to produce RNA transcripts. But once in a while, the nascent RNA, the 5' end specifically, is able to push the constriction so that it is removed and the tunnel becomes free. And that leads to a reduction of the affinity of sigma, and eventually sigma falls off. You have active polymerization. So here's just an example for why it is needed, why the whole process is needed. Here are just a few crystal structures out of the currently 20 existing ones from different species. I show only 4 because the rest are just almost homologous to each other, so it's boring. And you can see here basically the structure. This does not include the DNA. This does include the DNA in an open version. This includes the DNA also in an open version. This is when the bubble is open and NTPs are not incorporated. This is where NTPs are incorporated. And this is when it's in elongation, so the green subunit, the sigma, is removed. Now, the idea is that you can take existing crystal structures and morph them. Like, sorry, see the left one? It's okay. You can morph them and create movies, but they don't mean much. Here is the bubble opening. Here is the initial transcription. So you see domain movements, but this is just playing tricks with space and time. Who knows what goes on there? So being the skeptic that I am, and relying on actual experimental results, I'm going to tell you a different story. So we're going to focus on the open complex. And basically I'm going to tell you that this is not such a static transcription bubble. So actually there is a model that assumes that the 2 bases that belong into the hybrid actually are reeled into the bubble, so the bubble increases. And the identity of the first bases that are transcribed might be changed. So this was inferred from gels where you saw heterogeneity of lengths of transcripts. But what I did is basically I did an experiment where I had a library of 43 different pairs of distances where dyes are labeling the DNA either at a group above, upstream and downstream to the bubble. So when the bubble is enlarged, it is squeezed, or either both in the bubble. So when the bubble is enlarged, the distance increases, and I did this because I already know I expect more than one confirmation from previous results. So I need some logics in order to be able to classify the results. Those that belong to the scrunching coordinate distances will be anti-correlated with those that are belonging to the bubble coordinates. When one is high fret, the other is expected to be low fret. So here is an example of the experiment I did. I compared a distance at DNA, and I do it because in these measurements, no matter how much polymerase you add, you always have a fraction of unbound DNA. So this is the result. It's a single population, and according to the burst variance analysis, it has static heterogeneity, so it's really a single state, at least at that time resolution. But when you add the polymerase, and the polymerase opens up the bubble, but you don't add the nucleotide, so transcription doesn't start yet, what you have is the same subpopulation of the free DNA that is still with static heterogeneity, but additional bursts with higher fret, and they also have data that has higher standard deviation than the semicircle. You can see it more pronounced in other constructs. Here I use the construct that has you have to add into your calculations the actual free DNA subpopulation, but I also use the construct in which you have quenching between, so you don't have actually fret data, or here it's even more pronounced. You see you have nothing here. The dyes touch each other when the DNA is annealed, but when it's melted, then the dye get farther away from each other, and then fret information starts to appear. And this is nice because you can reduce the amount of states you fit to that specific measurement. But note that even in this case, when we focus just on the RNAP bound information, it's more than one population. More than one state for sure, this one population here. So I did 6 more. These are 3. I did 6 more, and the rest I excluded for activity reasons. I had a very nice activity assay that allows me to test 2 activities of the polymerase. Basically, if you have an active transcription, you would expect the polymerase to be able to open the bubble. And then upon addition of NTPs, you would expect the bubble to be transported with the polymerase to the downstream region of the gene. So you will have re-annealing of the bubble region initiation. So you will come back to the result that you had in the beginning, or at least with some efficiency. So these are the chosen ones, the active ones. This field usually uses these screenings. You start with like tens to hundreds of different distances. This is a lot of work, but eventually you end with tens that are enough to cover the whole, or enough reaction coordinates, so that the rest can be filled in with molecular dynamics, for instance. And these are just the data. So I just want to tell you what were the results. The results were the following. First of all, I compared expected distances. Expected from what? Expected from what you would expect for the dyes positioned on bases of a DNA that is fully annealed, in black, or in comparison to what you would expect from the known crystal structure of RPO, in red. So I compare theoretical to experimental, and what I get is that scrunching coordinate and bubble coordinate, indeed, no matter what, you have a nice fit of red to magenta, except maybe this point. And you also have, an additional population that is totally skewed out of the red, out of the magenta data. It is also statistically significant like that. And it's also anti-correlated between what you see in the bubble coordinate distances and what you see in the scrunching coordinate region. In the scrunching coordinate region, what you see is decrease in distance while in the bubble coordinate what you see is an increase in distance. So you can play around with your logics to start doing some assignments for those distances. These are one-dimensional distances. What you usually get is you have to remember that fret, and especially single molecule fret, using large dies, you attach those dies with long linkers. Those dies are by themselves large, so the distance that you measure is not between the attachment points of the dies, but between the mean positions of the fluorophores that are wobbling around in space around the attachment. But that's okay because you have means to compute the dye available volume, and assuming isotropy of movement of the dye, which is something I'm going to deal with in the next slides, basically you can build a map of possible positions of the center of the dipole, calculate what would be the mean position of that cloud, assuming the movement of the dyes is much faster than the conformational movements, and it should be like that. But there's a hidden assumption here that it's also faster than the lifetime of the fluorescence, which is not necessarily true. And then you start drawing lines and have sort of a fitting problem. So the first conformation, the one in magenta, fitted very well with the crystal structure of the RNA polymerase in RPO. While the other, again, it's not the structure what you see here in red and in green, which is fitted to the crystal structure in cyan and in yellow. It's not the structure, it's just the best fit, our best trial to the existing crystal structure. And it significantly deviates from it. What is the structure? Next thing to do is to throw all of this data into molecular dynamic simulations, starting from the crystal structure. But I can tell you already that at this time it wasn't so successful because you need a lot more restrictions in space. I'm working on that. I hope in the next few years there will be an actual structure. So, but this is just, you know, the best fit, and it's crappy actually. So the next step is to elucidate the structure, but I want to deal with this assumption that the dyes move faster than fluorescence lifetime. Fluorescence lifetime occurs at around nanoseconds, and the assumption assumes that the movements of the dyes is at the tens of picoseconds. Well, this is not exactly right, but I just want to point out to a new consortium, mostly European-based, of single molecule biophysicists doing single molecule fret, who got a sample, a DNA sample, just simply labeled at two positions. And since DNA, specifically bDNA, has a well-known structure, it's a good molecule to build on for standardization of fret. But all of them did the same mistake as I'm talking about, the mistake of not taking into account the fact that distance changes between the moment you excite the molecule and the moment the energy is depleted from the excited state. Well, biologists mostly would say, I got this answer a lot of times, well, this is in nanoseconds, nothing interesting happens in nanoseconds. But still, those are dyes, and those are freely moving, although on the other hand, they are large, so they are moving a large number of sheet of water molecules in order to do so. So the question is, how fast? Is it?
On DNA, are they freely moving, or is there some rotational anisotropy at this point, which might actually be very important?
The answer is very simple. It's part of my talk also. Yeah. The literature shows you that experiments of fluorescence and anisotropy decays on these specific dyes attached to DNA and to also proteins and many stuff, many other biomolecules are wobbling with a decay of around hundreds of picosecond to a few nanoseconds. So this is well within the window of fluorescence decay.
I guess once they're intercalated in the DNA, it might start becoming a little bit different.
No. In most of the time, they don't intercalate if you make sure that they are binding internally, they are covalently linked internally to DNA. If they're covalently linked to the 5' of the 3' end, it's a different story. The dye pi stacking starts to play a role on the edge, on the terminus of the DNA. So it's mostly just hydrodynamics of the movements. It's a large pi system. The smaller ones have tens of picoseconds relaxation. The larger one has hundreds, and if there are even a few nanoseconds. So, so this is the idea is that what you get in single molecule is still a time average, and you have a distance distribution underlying, okay, and also fast dynamics. But it's important for us also from the computational project, because we do want to go to molecular dynamics and use the Fret data as constraints in space. Molecular dynamics accept not distinct distances, but distance distributions. They accept the mean distance, which is the mean of the potential well for that coordinate, and a spring constant, which comes directly from the width of the distribution. You can see here the thermodynamic relation, of course. And we can elaborate more and more about the different models of distance distributions that you might find. I still stick to the Gaussian model. It's directly related to the potential well for that state. But here's the problem. And this problem was identified 40 years ago, but it's still highly ignored. The idea is that in equilibrium, you excite at time zero an ensemble of molecules, or in single molecule, you each time excite randomly a different molecule from the ensemble, it doesn't matter. And those molecules are distributed according to the equilibrium distance distribution. Because excitation is a Franck-Condon process. It happens instinctively. But with time, the distances changes. But the distances changes while the donor in the excited state, and you have a certain probability for the energy to be depleted. The probability to be depleted is just the first order probability dependent on the distance. So you have like an array, a spectrum of sinks for the excited energy of the donor that increase more and more. So, you know, thinking about this in a Monte Carlo type of approach would say, well, we can throw around probabilities and start to try to mimic the process, and maybe this could be a fitting procedure. So, as I said, this was already discovered and modeled by Haas and Steinberg, big guys in the Fret field. But the problem is that this Smoluchowski equation that is inhomogeneous due to this spectrum of depletion times is impractical. It's really practical for fitting fluorescence decays in ensemble where you have tens of millions of photons and the decays are really smooth. But such a complex model to be fit, and believe me, I tried, to be fit to single molecule fluorescence decays where you accumulate photons, but you usually end up with tens of thousands of photons or slightly more. The fluorescence decays are so noisy, you won't get anything out of it. So the idea is as following. Remember that you have basically a time series of photons for each molecule, but most importantly, you know that all of those photons are coming from the same molecule. Therefore, it's a time series, it's a time-dependent series where you have sparse data, because underlying you should have some sort of distance trajectory. Okay, but what you have is distinct times in which you have photons. And if you look at two consecutive photons and think about a pulsed excitation scheme, where you have a pulse here that produced this emission, and then many other pulses that didn't introduce any emission, and then another pulse that introduced the next photon, what you have is two time scales. The time between the first photon and the excitation that led to the second photon, and the time between this excitation and the second photon, that you can play around with Monte Carlo using stochastical differential equations of the Ornstein-Uhlenbeck process. So imagine that you have this distance trajectory. It doesn't matter whether it's really that one. It's just the statistics of the distribution and the dynamics that led to it. So this is the Ornstein-Uhlenbeck process that we heard about a lot. But the problem is that in order to use it in Monte Carlo simulations, you have to have infinitesimal time increments. That's kind of a computational problem. But it can be solved if you have a direct update formula where it doesn't matter anymore what delta t you use. You can use as long as you want. You can get, with the noise of the randomicity of the Monte Carlo process, a good estimate of the next distance dependent on the diffusion and on the variance, which basically depend on the correlation time. So the idea is the following. For the time between excitation and emission of a photon, you have this Monte Carlo simulation running. You start at time 0. You ask what's the distance. You ask what's the probability to be de-excited. And then you ask, was it de-excited? No. You go on and produce the next distance. Yes. You save that data. And this is the probability. And then after saving the data, you convert it to fret, because this is the experimental observable. And you do it with another probability function. This is the first-order mechanism for the efficiency, basically asking if the excitation occurred, what's the probability for it to be because of a transfer of energy and not because of spontaneous emission. And then you make the data a little bit dirty, because it is actually dirty, because you have also leakage of the donor fluorescence into the acceptor, you have direct acceptor excitation, you have inconsistency between the detection efficiencies and the quantum yields of the donor and acceptor photons, so it's not one-to-one. And then you ask, was it eventually a donor and acceptor photon? If it was an acceptor, you label it, then you ask just another random process at that period of time. Was there blinking? Blinking is a little longer process, but you have to take that into account. If it was, and the process just relies on the rate that you have in the model, the rate of blinking, then it turns out to a green photon, because if the acceptor is blinked, then you only have a green photon because only the donor is active. So I will skip that. Basically, these are the simulations. But eventually what I want to at least try to convince you that instead of fitting, why not have a really good model with a really good simulation, hopefully with enough priors, to try to fit the simulation results. So in essence, the best situation would be that we would fit each photon, experimental one, to each simulated photon, but we can't reach that state. Maybe we can in the future. There are a lot of variables that make it hard. Instead, we have a lot of different statistical histograms that we can accumulate. The ones that are usually used in the experiments are those fret histograms that are simply an accumulation of instantaneous fret for different molecules. But you can have a global fit of the simulation results where you also rely on photon statistics. These are just the fluorescence decays. You can take into account also FCS coming from your data, mean nanotimes, you name it, and you start to constrict your result into the actual one with small error. And indeed what you see that is the fit is good. And indeed what you get is that, as we expect, what you measure in fret is not that. And this conference is all about reverse problems. What you measure in fret is the distances that were caught when you had depletion of the excited state of the donor. But this was changing while you were waiting for the depletion to occur, starting from the excitation.
Okay. I'm sorry, Eitan, but that's the lunch. But can you just conclude, like, in just a couple of sentences? Thanks. Okay.
So skip. This is why I prepared that button.
Skip.
So as you see, we have capabilities, but they are now even more improved for accuracy of getting the data on conformational states, at least the one-dimensional ones. If we have enough of them, we can start to construct structures, combining these results with simulations. The DNA bubble transcription initiation. So transcription start site selection might occur by spontaneous scrunching of 1 to 2 bases downstream to the bubble. And this might explain the heterogeneity in the transcript lengths you have. So it's important to take into account the distance distributions that you get, but the data is much more complicated from the theoretical point of view than just thinking about it as a simple molecular ruler. And that's basically the future goal. I have part of the data now. So the outlook is to use this technique in many different ways to study the structural basis for a lot of different conformational states that are sparse, that you don't see them even in cryo-EM, to study even things with more complicated photophysics, like doing multicolor FRET in order to study allosteric synchronization between different domains of proteins. But eventually, it would be nice, after having a dictionary of signatures for different conformations, to throw those labeled molecules into the mix of the bacterium. You can do it with many different techniques. And since we have the signatures, in the sea of what you have in an imaging experiment, now you will be able to better identify specific states that you already had in your dictionary in vitro. So, it's important to mention, especially nowadays, that all of my data, all of my code, all of my raw data, all of my process data, everything is up in the web. And there is a lot of code going on for the WISE lab, and now I'm also joining as a different lab for open computational tools for single molecule spectroscopy. It's Python and GitHub, so it's free to use for everybody, free to modify by everybody. You name it. I want to thank especially my previous PI, Shimon Weiss in UCLA, and this postdoc of Shimon, Antonino in Gargiola, who is now in Milan looking for work. He's a very good scientist. I really recommend it. And my new lab in the Hebrew University, and the rest of my collaborators in the Institute of Jacques Monod, in Rowan University, and in Boulder. And with that, I would like to thank you, and I hope it was interesting enough.
Thank you very much, Eitan. And now let's go for lunch and dinner after.
All right.